All right, the final part of this chapter is going to be on the one gene, one enzyme hypothesis. So this is the result of a lot of work from George Beadle and Edward Tatum, who showed that genes are directly responsible for synthesis of enzymes. So they used this studying nutritional mutations in bread mold neurospora caused by lack of enzymatic activity. An experiment led to the one gene, one enzyme hypothesis. So I think this will seem very obvious to you. We kind of build upon these experiments, and now it just kind of is the fundamental assumption of how things work. But this is the original experiments that allowed us to conclude to build this intuition. And so what they did is they analyzed more than 2,000 strains that had been mutated and with light in order to produce mutations. And then they identified a bunch of these mutations, these mutants that were unable to synthesize certain key vitamins or amino acids. And so this should bring you back to the chapter 6 on bacteria. So, the bacteria require the ability to create certain chemicals. So it requires the ability to create amino acids, it requires the ability to create certain vitamins. And so if they have all of these enzymes, then they're able to take minimal media and they're able to produce every single thing they need from that very simple media source. But if they have a mutation, they might lack some sort of enzyme that is responsible for that pathway that is required for production of some amino acid or something like that. So, all they did is they just mutagenized a bunch of different strains, and then they incubated the strains in rich media in order to get, a large source of the same strain. And then they transferred this to a large number of different types of media. So they might have transferred it to complete media, so, rich media that contains everything that you need, and so you see it's able to live. But because this mutation, this mutant animal, this mutant bread mold spore doesn't have a key enzyme, it's unable to live in minimal media. And then you can just supplement with all sorts of different amino acids, vitamins, and you can find where exactly this mutant is defective, which part of the pathway. So we see if we supplement with arginine, we're able to the spores are able to survive. And so they use complementation to identify independent genes. And so the reason this was not done in bacteria, the reason this was done in this bread mold, is because they also wanted to have the ability to create diploids. And these fungi actually can either reproduce as haploids or diploids. It's kind of like a special thing that they're able to do. And so this was important to be able to perform complementation tests that we covered in Chapter 5 in order to determine how many unique genes were affected. And so they use complementation tests. So if we had a bunch of different mutants that were unable to grow unless you supplemented arginine, you could determine whether or not they were in the same gene or not. And so if they were in separate genes, then if you fuse these haploid cells together to form diploids, now these diploids were able to survive and produce arginine from minimal media all by itself. So these heterokaryons, as they're called, are able to grow without arginine because they have one good copy of ARG1 and one good copy of ARG2 because we've created these diploid cells. So they identified a large number of mutants, and they categorized them into the same gene pathways. And so now they were able to take their knowledge of biochemistry in order to further break down what these mutants were lacking. And so they could do this because they knew the biochemical pathway by which methionine was created. And so there's a large number of intermediate molecules that need to be created in order to finally produce methionine. So homoserine is converted into O-acetylhomoserine, which is converted to cytasiathionine, which is converted to homocysteine, which is converted to methionine. And in each case, there's a unique enzyme that is required for this molecular reaction, this chemical reaction. And so they knew that there was 4 different enzymes that were required for this pathway to eventually create methionine. And so if you had a defect in any of these enzymes, then you would be unable to produce methionine. But they reasoned that each of these genes, so there would be different genes, we'll just call them ARG1 to ARG4, each of these genes, might be defective and you wouldn't be able to grow methionine, but you could rescue them in different ways. And so if you have a mutation in this gene where you're unable to convert homoserine into O-acetylhomoserine, then you could rescue this, we know, by adding methionine. So if we just add methionine to the media, then these cells are able to grow just fine. But we also can rescue them by adding the upstream component of the chemical reaction pathway. So if we add this molecule as well, the cells are going to do just fine, because even though they can't transfer this homoserine and produce this, they're able to take this from the media, and then they have the intact ARG2, ARG3, and ARG4 to follow this along to eventually create methionine. Whereas this mutation right here, if you provided it with O-acetylhomoserine, it would unable to rescue because it could not change this into the cystathionine. And so this is the kind of logic that you use in order to place these mutants into pathways. And so here you grow it in minimal, or you supplement with this, or you supplement with this, you supplement with this, or you supplement with this, and then you get a different range of mutants that grow in each of these different places. So in the normal strains, a given amino acid was synthesized by cellular enzymes, enabling them to grow in the absence of that molecule. In the mutant strain, a genetic defect in one gene prevented the synthesis of one protein, and so the conclusion was a single gene controlled the synthesis of a single enzyme. So it was this reaction, this experiment right here, which showed that each of these could be rescued by different molecules, which imply that each of these enzymes were encoded by a single gene. So each mutant had a single one of these enzymes ablated by the mutant. There was a loss of function, and so that was one of the primary conclusions of Beadle and Tatum. And so this was referred to as the one gene, one enzyme hypotheses. And then a final example of this, one of the first diseases to be discovered connecting alleles to protein variation was sickle cell disease. And so sickle cell disease is a disease that we've talked about earlier, where a mutation in hemoglobin leads to a ability of the hemoglobin to clump together to form these filaments, and these filaments then end up warping the red blood cell into these sickle cell shapes. And so this was a disease that was known to be genetic, it was known to be inherited, it was known to have this effect on red blood cells that could be seen just by taking blood samples and looking at under the microscope. But at the time, no one was able to know what exactly was causing this disease. How is the genetics leading to this change in this blood cell? And so this fingerprinting was actually able to be used to show that there was some sort of change in the hemoglobin protein. And so they were able to do this because the genetic change was a change in a amino acid from a glue to a valine. And so the way they determined that is they took the hemoglobin protein and then they cut it with specific peptidases that could cut protein at particular amino acids. And so if they used one that cut at valines, they were able to show that the size of the protein fragments were different, because in one case you were able to cut, and then in the other case you were unable to cut. And so the fragment sizes of the hemoglobin were different, and they correlated with the disease, whether or not they depending on the genetics. So this was the first human example of kind of the one gene, one enzyme hypothesis, which showed a connection between the genetics of a disease with the effect on the protein sequence. And that led to changes in, for example, red blood cell, and then obviously the symptoms of the disease that are caused by that. So that is the end of chapter 14, and that covers translation and proteins.